脑人言。
各位老师、各位同学，大家好！感谢大家抽出宝贵时间来参加这一期的老人言论坛。那么我们这一期有幸邀请到了就是加州理工学院的聂斯博士。那么聂斯博士本科毕业于华中科技大学，博士毕业于中国科学院生物物理研究所。那么其后他加入了这个加州理工学院。那么聂斯博士今天带来的报告题目是这个核孔复合物的结构与功能研究。那么我们把时间交给聂博士啊。
谢谢小田我今天因为有很多的一些专业术语，我可能不太清楚，就是很短的时间把它就是翻译出来，所以我今天可能会用英文来讲，我会讲稍微慢一点，然后如果大家有疑问，随时欢迎打断提问。Thank you for having me here today. And I will say good afternoon, good morning, and good evening, or sometimes good afternoon, everyone. Thank you for having me here. Today, I'm going to talk about the structure and the function of the human nuclear pore complex. I guess here, everyone is familiar with the nuclear pore complex. We all learn it on the textbook. It's actually a transport channel that embedded in the nuclear envelope. It's like regulate all the materials exchange across the nuclear envelope. It's discovered in 1915s when people tried to use electron microscope to observe the nuclear envelope. They found there are pores, and then it opened this nuclear pore field. And 9 years later, people found, okay, it's not only pores, but had some protein structure. They named the protein as nuclear pore complex. On one hand, people always use electron microscope to analyze the structure for the nuclear pore. 1967, this year, people found that the nuclear pore actually has an 8-fold symmetry. In 1993, people were able to establish the first EM structure for the nuclear pore, but at that time the resolution is really low at 80 to 100 angstroms. With technical development, this resolution get higher and higher. Besides, people also found that nuclear pore complex also has some portion stick out. They found there are cytoplasmic filament on the cytoplasmic phase, and also they found the nuclear basket on the nuclear phase. All these electron microscope study establishes the overall structure for the nuclear pore. It has a base anchored in the membranes, and it has a cytoplasmic filament and the nuclear basket. On the other hand, people try to identify what's the component for the nuclear pore complex. They did a very interesting thing that they get the antibody that target to the nuclear pore, and then use the antibody to pull out and identify the first protein. They call this protein NAP1. And later people then use protein-protein interactions, pull downs, mass spectrum were identified. There are 34 different proteins belong to the nuclear pore. They name this protein nuclear pore rings. These nuclear pore rings exist in multiple copies of 8, sometimes 8, sometimes 16, sometimes 32. In this 8 copies were established 8-fold symmetry structure. Generally, this nuclear pore ring can assemble into several major subcomplexes. The first one, also the well-studied one called cold nuclear pore ring, is sit at two sides of the nuclear pore. Besides, there are inner ring nuclear pore rings. It occupied the center of the nuclear pore. These two subcomplexes assembled into a symmetry core of the nuclear pore, provide binding site for the cytoplasmic filament and the nuclear basket. Besides, there are pumps called transmembrane nuclear pore rings. It can anchor this nuclear pore into the membranes. It's very this protein very conserved from human to the from the east to the human system. This nuclear pore ring contains a lot of filaments, and the glycine motif. These motifs are quite like flexible. By projecting this like flexible motif into the central channel, the nuclear pore complex are adaptation or like diffusion barrier. We also call this size selecting barrier. This barrier like prevent the molecule bigger than the 40 kilodalton passive go through, but allowing the smaller molecule go in and out freely. To overcome this diffusion barrier, nuclear pore complex use the importing and exporting this transport to carry cargo bigger than the 40 kilodalton pass through the nuclear pore. This process is regulated by a small run GDP called a small run GDP. So like the importing will recruit the cargo in the cytoplasmic and carry it through the central channel. When they arrive the nucleus, it will disassembled by the run GDP. The exporting will do opposite things. It were assembled into in the nucleus or disassembled in the cytoplasmic by run GDPs. By doing this, nuclear pore complex actually acting as the surrogate that controlled all the materials across the exchange across the nuclear envelope. It's definitely important for the nuclear for the gene expression is crucial for all the cell activities. People always wonder how the structure look like, wonder use this structure to guide the function study, but how this structure can be determined. This nuclear pore complex is huge, even though it only has like 34 different proteins, but it has 1,000 protein, is dynamic. So, and also flexible. So people like our lab use a divide and conquer approach to get the structure. What's the divide and conquer means? That first step, you determine the high resolution structure for the nuclear pore rings or the nuclear pore ring complex. And the second step, we're fitting this structure into the like a creatine map. And this fitting only according to whether this model fit this density. And then the third step, you will need to do some like interactions to validate, to add something that doesn't stay in the electron max fitting. 2016, our lab will use these divide conquer methods, generated the first near atomic composite structure for this symmetry core. This structure shows that this symmetry core has 3 rings. 2 rings of cold nuclear pore rings sit at 2 sides of the nuclear pore, definitely in inner ring like sit inside. This structure get improved like this year by another they provide they introduce more details by solving a lot of structure. All the structure belong to the inner ring nuclear pore rings 155, 93, and 25 and 188. And then use this structure, they like finish the characterization for the symmetry core of the nuclear pore. And what left over is the asymmetry nuclear pore ring complex, the cytoplasmic filament, and the nuclear basket. That is one of my focus, which is cytoplasmic filament nuclear pore rings. People use like immunogold to identify the position for the nuclear pore rings. They were identified 9 different nuclear cytoplasmic filament nuclear pore ring exist at the cytoplasmic phase. The first identified nuclear pore rings are 88 and 213, and then there are like another 16. There are 8 are quite 8 nuclear pore rings quite conserved through the fungal to the human system. We first use like a backcountry interactions IC to analyze what's the interaction network between these nuclear pore rings. We found that these 8 conserved nuclear pore rings can form a conserve module architectures. The key is 88 to 159 and NSP1. It can form a it can form a complex through its coil code and then provide like binding site for two major mRNA export transporter called RIL1 and GLI1 DDX19 and form a very conserved structure. Besides this like eight conserved nuclear pore rings, there is another one called lab-358. Is one of the largest nuclear pore ring exists in the nuclear pore complex. It has 3,000 different proteins, and previous studies show that this protein can form a 36 nanometer filament under the microscope, and this like filament lengthwise is very much the original observed filament of the nuclear pore. So, and then later people like deleted this protein, found this filament disappeared. Then it established that the 358 actually is the primary component for the cytoplasmic filament. We also did the interaction analysis of the 358 with other nuclear pore rings. We found that since that 315 stick out, it doesn't have any necessary contact with this like another module. Thus, it establishes that at the cytoplasmic filament, there are two module structure. One is this like 8 conserved module CFNC, and one is 358. So to identify the structure for this module structure and also the entire structure, we use divide conquered methods again. The first one is 358 structure determination. We were able, so like 358, it shows that like 358 N-terminal region have anchored is towards the nuclear pore, but the C-terminal extend away. This indicated that N-terminal part is the anchor site for this 358. So we first were able to generated the proteins. There are two fragments can work, one short form, one longer form, but these two proteins like behave a little bit different in the solutions. With a longer form, it doesn't stable in the lower salt solutions. It's crushed out, and even with high salt concentration, high salt solutions, the protein began form a tetramer and pentamer in the solution. With this like short form, since is stable, but with protein concentration increase, the protein began to form a dimer in the solution. With this protein, that we start to do the crystallization structure determination. But after we extensive efforts, we didn't see any crystals. So we decided to use a synthetic antibody as crystal chaperones. We collaborate a lab in the University of Chicago. They help us. We give them proteins, then they help us to generate select the antibody from their feed pore, and use this we already use this antibody to solve two major structure. One is the CNT, another one is the cold nuclear pore rings. We were able to generate 62 different antibodies. I tested their behavior over demonstration with 358. I found at least 9 different antibody can form a very beautiful complex with 358. Use one of these antibody, I could generated a 3.9 instruments crystals and get a model. But some people may know that with this resolution 3.9 is very hard to determine the position. It's very hard to see the side chains then it will be very hard to assign the sequence into the map. So we will generate like thymine methionine mutations in every helicose, and by determining the position for these proteins, for this amino acid, we could determine where the protein sequence in the map, and finally generated a very accurate model for 358. In this in this in the asymmetry unit, in one asymmetry unit, there are two copies. These two copies of 358 interaction have very extensive interactions. It will generate a problem, which is there are two conformations. If you see this way, the N-terminal extend, you will see like a open conformation is X-shaped S-shaped conformation. But if you look at this way, that this TPR region where like N-terminal region were falling back, it form a closed form. At this stage, we cannot tell which conformation is a real state, is like crystallizations. But later we can tell, like there's other evidence can indicate which conformation is real. So after we got this like structure, we want to determine the position for this 358 in the nuclear pore complex. We use two different way. One way is try to dock the structure into a format that everything doesn't sign anything. Another way is that you take out what already know and only search the model in the leftover density to see whether these two methods can give us very like similar results. If the results show yes, it's similar, that's we can see 5 copies of the open form 358 can dock into the cytoplasmic phase of this nuclear pore. So we can see that this N-terminal region can fit the density quite well, but we try to do the same thing for the closed form, but this crash out. This this approach indicated that probably in the solution, the open form of 358 is the real state. When we look closer, we found that like a full copy of 358 wrapping around the stick of this called nuclear pore rings, and with one copy of like 358 linked at above of these 4 copies. And we are wonder why there's like 5 copies. What's the factor to determine these oligorate states? We look closer to the like sequence. We found that there are coil-core domains. By determining the structure, crystal structure of this coil-core domain, we found that this coil-core domain can form a tetramer, and also it form a tetramer in the solution. And this like element can induce 358 form a pentamer and pentamer in the solution. When we try to introduce the mutation to break these tetramers, the protein no longer form an oligorate state, oligorate state in the solution. This experiments establish that this coil-core domains is the trigger for 358 oligorate state. After that, like I go ahead to determine all the remaining of 358 structures. 358 besides the N-terminal region, the coil-core domain, it has 4 run binding domains, run GTP binding domains, and has multiple run GDP binding domains. I get like 2 angstrom resolutions. With all these models, all these structures, I would able to generate the 315 model at the cytoplasmic phase. There are 5 copies interfaced with the cold nuclear pore rings introduced by these like oligorate states. 350 that can form a bundle projecting into the cytoplasm, provide the binding site for bunch of the run GDP and other and run GDPs, and like a kind of provide binding site also for other cargoes. And at this stage, we can tell one row for 358. So remember, I like mentioned earlier that this cytoplasmic filament cytoplasm transport is regulated by run GDP. But we look at 358, it has 4 run GDP binding domain and multiple run GDP binding domains. A key component is E3 ligation. It can bind to run GDP, and then run gap. It can like hydrolyze this GDP to GDP form. Then at the cytoplasmic, there are huge amount of run GDP, which is important for for this release cargo from the transporters. Is kind of 358 acting as a run sink, provide increase the local concentration for run. And next, we try to determine that what we see in vitro in the cryo-EM docking is real. We try to identify the minimal and necessary fragment for 358 in the cells. We divided the cells into 2 parts. One part like N-terminal part and C-terminal part at different positions, and try to see their localization. We found that the N-terminal plus the oligoride state is necessary for 358 targeting to the nuclear pore. When we like further cut down the protein no longer localized to the nuclear ring, and also this oligoride state is important. We introduce this like oligoride mutation to this protein. The protein be no longer localized to the nuclear rim, and also we try to introduce mutations that break this N-terminal and like a cold nuclear pore interface. Is this protein also lost its nuclear rim localization? This like cell experiments adapt that the N-terminal plus the oligoride is important, also validate our EM docking result. After we sign the 358, there only one leftover density is kind of rock state. Consider its position. We reason that is the position for the CFNC. Could we do the same thing? The answer is no, because there are 2 structures we don't know. Which one is the core domains, one is 88, 98, and 214 binding. We're able to generate the crystals for 88 and 98. We're able to get the structures and compare this structure between different species. We found that this like 88, 98, 214 are quite conserved. And then with this structure, we will have only one left over, which is this coil cord. This time, since I try a lot of crystallization, it doesn't work. And also this antibody, we also generated antibody to help, but this time antibody doesn't help. We also try to do the like cryo-EM single particles. We can see filament, but doesn't work. How we get the structures? We figure a different way, which is use the we use this protein that's NAP62. This coil called NAP62 is also a component for the channel nuclear tremors. This structure already solved. In the structure show that it also have the same coil cord like arrangement, the first coil cord, second, and the third one. We compare this like sequence. We reason that this coil cord domain should have the same structure shape as this channel nuclear poring chambers. We get a model from this channel, and luckily on exactly that this structure can dock into this only leftover density. By putting this coil cord in, we are then like very easily to put 88 and 98 because the n-terminal region is there, and then the 214. The model fit the density quite well. And with this docking, we found a very key point that since this coil cord region is quite close to the NUP 96, NUP 93, which is an inner ring nuclear inner ring complex component. Consider this like position where like the first this there is n-terminal region and this side, and there's c-terminal. Even though there is no density for this region for 93 C-terminal, but we consider the distance. We reason that it's probably the anchor side for this like CFNC. The answer is we test their interaction in vitro. We found that indeed this 93 could interaction with human CFNC. It can interaction with this coil cord and also can interaction with this four complex. This is a complete 6-8 proteins complex. But there is after we figure it out, we then ask ourselves, is that similar? Is that's also conserved in the fungal system, because that is the human system, and all nuclear pore complex is quite conserved from east fungal system to the east. We test these interactions in the fungal system cotomial, but we found that no, for the CFNC anchor site is different in the fungal system. We get this NIC96, which is a component, which is a human 93 homolog. There's no interactions, and we ask ourselves, okay, where is this protein actually anchored in the fungal system? At the cytoplasmic phase, besides these cytoplasmic filaments, there are called nuclear porins. Like obviously, we will test whether these cytoplasmic filaments there is any interaction with cold nuclear porins. We get all these like small fragments exist in these cold nuclear porins and found one protein, one small fragment at the C-terminal of 37 could interact with this coil cord. And this interaction could bring this cytoplasmic filament to this cold, to the cold nuclear porins. At this stage, all our biochemistry study and structure study established the entire structure for the cytoplasmic phase. And we next wonder what's the function for this cytoplasmic filament. So the previous study showed that this cytoplasmic filament could had some roles in the mRNA export. As we may all know that mRNA first synthesized in the nucleus, and then with this nuclear pore transport is where transport will go to the cytoplasmic phase. But there is a very key step at the cytoplasmic phase. The CFNC we're remodeling this mRNA, the mRNA by remove the mRNA, remove the transporter factors. By remove these transporter factors, it will prevent this mRNA moving back to this nucleus. And since this CFNC has our lab and also our lab already figured out that the CFNC have heavily wrote in that, but there's no study about 358. So we first want to test whether 358 have some wrote in this mRNA export, the first thing to test is that whether 358 could interaction with RNAs. We get all these proteins and test their interaction with single RNAs and also the double-stranded RNA. We found that yes, indeed, 358 could interaction with this mRNA. Is that mean that 358 indeed play some function in the mRNA export? To analyze that, we want to establish one cell line that we can delete 358 and to compare the deletion and a control cell to see whether that 358 could involve could play something play some functions in the mRNA export. We were able to establish inducible lab 358 lockout cell with Marie-Dassault lab. They insert like AID in front of like 358 with oxygen tree, the protein can degrade in 3 hours. We test these cell lines with immunofluorescent, also Western blot, found that after 2 hours, the protein already like under detectable. We cannot see also 3 hours totally gone. With this cell lines, we then to identify, try to see whether 358, in fact, mRNA export, we do one thing. We do one IC called pose and chase. We first label the protein, label the RNA in the cells, and then monitor the RNA's position in the cells with or without 358. After we see this result, we think that 358 control cells and 358 depletion cells seems have very similar trends of and a similar RNA localization. And we like analyze the data, see that same 350 doesn't affect mRNA export. To avoid like experimental bias, we also introduce a laser cell lines with 160 knockout, which is a component for Y component. With this deletion, you will expect the nuclear pore complex get disassembled. The protein with this cell lines, we see a very we see there are mRNA defect, which means that our experiment doesn't have any problems. It just indeed 358, the same doesn't affect mRNA export. And let's try to see what happens when this mRNA get export. Is there whether 350 also affect some translation? We express 8 different reporters in the cells, found that with 350 duplication that these reporters expression never get a dramatic decrease, which indicated that 358 play a general role in the translation of the translation, but it's like first established there are something functions of the nuclear pore in the translation, but we don't know yet what's the reason. Hypothesis is that because 358 could bind to the RNA and then it will hold the RNA there. It give the CMNC enough time to remove the mRNA export factors, or their 358 actual interaction with some mRNA affect the mRNA handover to the translation machine. Is there any interactions? We don't know. It needs some study. That's interesting part. Another thing is that we always wonder that how we got the structures, how this what we can do with the structure. Nuclear pore complex is a surrogate, is involved in a lot of cell activities, also disease. One of the key things that it definitely involved in these virus infections. About also like recently very popular, the SARS. We found that like SARS actually can like SARS-1 components can interaction with the like cytoplasmic filaments, which indicated there's some like some route. We are like doing a lot of like assays in the lab now. Hope we can get something out. And now I like want to think our lab. This like project, you can see that I only show a little bit of like very small portion of the data here. We have like 300 page of like data. People in the lab working very close and hardworking, and also want to like our collaborate lab generated the antibody and generated the cell for us. And then like I'm happy to take any question you may have.